Okay, so as I was saying, make sure you check for grammar and spelling throughout your lab report. Then here, methods and materials is literally just the equipment that was used and what it was used for. You need to write this in sentence format. Do not use bullets. You will lose points. Then for your data, this is just your tables. You do not need to discuss anything whatsoever in the results. Okay, this is just the two tables, abiotic, biotic. Then your discussion. Now here's where most students struggle, which is why we've given you a number of resources and helps for writing this section, because this is where you have to work through that thought process. For example, down here you're going to discuss the results of nitrate in relation to the stream environment, okay? It says compare the results to the acceptable concentrations. Well, what's an acceptable concentration? Where do I get that? Your lab book is where you get that, in Stream Analysis 1. There's a discussion on nitrate. It said the permissible level for drinking water is 45 milligrams per liter of nitrate. That means there's 45 milligrams of nitrate chemical in 1 liter of water, and that is safe to drink. It won't make you sick or have any type of consequences. However, if you have more than that, there will be consequences. You could get sick. It's considered unhealthy to drink. And less than that is considered just fine. You can drink it. So you need to look at your data, look at the average specifically of your data, and decide, well, is my average above or below 45? If it's above, it's dirty, don't drink it. If it's below, it's just fine, you can drink it, okay? And that's what you're gonna do for all three nutrients, okay? Now with dissolved oxygen, you have to consider the saturation point as well as whether or not aquatic organisms can survive. Now in the book, it talks about how many milligrams per liter you need so fish can breathe, essentially. And then there's also a chart for saturation. Saturation is just how saturated the water is with dissolved oxygen, and that is dependent upon temperature and flow rate. That's why up here, I've asked you to describe the relationship between flow rate and dissolved oxygen, which you will gather from your reading, okay? Humans in nature can offset flow rate and dissolved oxygen. Think about, what would happen if it rained a lot, okay? What would happen if humans built a dam? Things like that, okay? Don't mention pollution. The only way that pollution is really going to affect flow rate of a stream is if there's copious amounts of pollution that create a damming effect. Nutrients and things like dirty water, that's less likely to create pollution unless it's, a extreme scenario where there's significant algae buildup and the algae works like a dam. It's a lot of times students talk about pollution here. This is not where you talk about pollution. This is just where you talk about flow rate, okay? Down here in your results for the nutrients, that's where you talk about pollution and what the environmental impact of those are. Then don't forget to talk about the watershed, how diverting the natural course of the watershed can influence the aquatic environment. Make sure you define watershed in detail and don't just give like a 1-sentence answer. Actually develop this. Notice that it's 8 points, okay? And then, of course, your references. Now, you, of course, have to have your citations, all your resources that you used. Please do not use things like ask.com or Yahoo Answers, okay? Those are not reputable resources. It's very important that you use a viable source. Use something like OwlNet through Purdue University. Use your book, your textbook, your lab book, okay? You can use scientific web journals as long as they are legitimate, okay? They have to be an actual scientific journal. You can use Water on the Web, which is a great resource for water concentrations and information. You can use the EPA, the USGS. All these things are acceptable, okay? But don't just give me, like a Reddit, for example. It's not credible. And then you have to use parenthetical references. Now, what that means, okay, is when you're up in your introduction and in your discussion and you're using a definition or you're paraphrasing, you have to put in parentheses what that source is. You don't need the full citation. You just need an abbreviated one. And what we use is author, comma, year. Okay, so if you're going to use the lab manual for the nitrate definition, for example, you would then, in parentheses at the end of that definition, write in Geary, comma, what was that? 2016, I think is when we published. Okay, that way, when somebody comes through and they read your report, they can go, oh, I want to know more about this thing. And then they go to your reference page and they look for author Geary and their 2016 article. Okay, so make sure you use that. You will lose credit if you do not do parenthetical references, and quite honestly, they're very easy to do. So just pop them in when you're doing your defining and paraphrasing. Okay, this is the document that I get out to even the playing field so that I am grading everyone precisely exactly the same. I go through and I make a checklist. Was it included? Was it not included? Or did you kind of include it? And you will be graded precisely according to this document. I highly recommend taking this out as you are writing your sections and checking off for yourself, making sure you did these things. Okay. Okay, so now that we're done with the rubric, let's look at the remainder of material. So here you are in your folder. Laboratory, stream analysis, test equipment lab, and stream analysis lab. Okay, so you're going to follow your week 1, 2, 3 guide, all right? And work ahead. By all means, work ahead. It's in your best interest to do so. And then down here, okay, in the additional resources first, let's see. We have just some helps in here, okay? How to do a website citation, how to create and share a Google Doc if you're going to do Google, all right? How to create tables. If you don't have Excel, you don't need it for this. You can insert a table in Word just by doing this. Let me show you. So let's just pick one of these. Okay, so if you wanted to insert a table in Word, you go to Insert. I'm going to put it right here for now. Table, okay? And then you just highlight however many boxes and rows you want, and boom, your table appears. And then you can just type in it, okay? So you don't need Excel. You don't need to copy and paste from Excel. You just create your table right in your lab report. But if you don't have Word and you don't have a good program like that, then you can just use Google Docs, okay? And it'll do essentially the same thing. And here are some helps to assist you in doing that. Then, okay, down here in the bottom, here's you resubmit your hypothesis, and then we have some more supplemental material. Now this is what is in the directions, okay? It's just all the videos have been broken out for you, so you don't have to refer to those directions if you don't want to. So here we go, okay? Here are the directions which I downloaded in the beginning, okay? Which, just really quick, if you don't remember where those are, we'll go back to the beginning, and they're those. Okay, so download those. Those are this document here. There we go. Okay. So this is your lab report, right? This is going to tell you how to do things, okay? What you need to be paying attention to. Read carefully through this. It's pretty straightforward, okay? So because you're not actually going down to the stream to do this experiment, you're going to watch the videos of someone else doing that, okay? And these videos kind of explain what you would be doing in the lab, for example. And those videos, okay, are broken out and placed in this folder here, okay? So here are all those videos that have been embedded into this file, okay? So that's all the differences, okay? So you can get them from this document, or you can get them from this folder, okay? I've also partitioned out the data, so if you don't want to grab it off of the online directions, you can just grab it from the data file, okay? But here is your data, okay? This is what you're going to use for nitrate at location A, okay? So here's a map of the stream, and here's location A, location B, location C, okay? This is where the class would have been obtaining data. Here is the satellite image. You can see this is Utica, Proctor Park. This is the creek that runs through it. I highlighted it, okay? This is just showing you were we to go to the stream. This is where we collect data. Here is a data set from that stream in those locations. So site A, 35.4 for nitrate. Site A for phosphate, 0.12, okay? Site B for nitrate, 26.7, okay, and so on and so on. You are going to take this data and put it into the data table for your abiotic results, okay? You're also going to have a column for your average. So you can, if you want to, just type in, okay, well, what's my average? So I have that. You are going to add up these three numbers and divide by 3, and then you're going to get your average. When you are doing your discussion and comparing your results, you always discuss to the average, okay? And so you just take all this data and you plug it into the data table. Same here for biotic, okay? Here are the critters you found. I'm using air quotes. You found these in the stream, okay? Use these critters. You can use the same image that I've used, okay? But you have to figure out who these critters are. What are they, okay? Now, if I were to create a data table, then I would be typing in, okay, nitrate, phosphate, all the other data that I showed you, okay? And I'm actually going to put these over one, okay? So you would have your, you can put your averages down here. You can put your averages over here, whatever you need to do, okay? Label this kind of thing, and then just take all that data from up here, okay, and plug it into your data table in your lab report. I'm just using this data table that we inserted, okay, for example. Now, how do I figure out what these critters are, right? I don't know what that is. Okay, well, there's files for you to look at, okay? So here's your stream life, 1 and 2, macroinvertebrates and microinvertebrates. You got bigger, see it with your naked eye, and you got littler, see it with a microscope. So you're going to open those up, and all of these critters that are in your biotic data set, they're on there, I promise. Go to that file right here and find them, find their name, okay? And then you're going to put that into your biotic data table by copying and pasting, okay? So that is what you need to do, okay? So really quick, I will try to summarize in the little bit of time I have left here, okay? Go to stream analysis, read the directions, okay? Watch the videos, follow this weekly guide, work ahead if you would like, okay? Download these guidelines, absolutely follow the rubric, complete your scavenger hunt, develop your hypothesis, and then write your lab report. Use the data here. These are the videos again. This is a Google Doc that contains your abiotic data. Okay, so it's all the same. It's just in a number of different places depending upon how you like to work. All right. Take all of this data, summarize it into the abiotic data table. There is an example of this in your lab manual. Take your biotic data, copy and paste it, figure out what those critters are using these files, okay? And summarize it into a data table, into your lab report. And of course, don't forget your references. So I hope these videos were helpful. Email me with questions, okay? But that is what you need to be doing, and that is what all of these different files are for, okay? That is all. Please do not wait until the last minute, and work ahead if you can. All right.